We study the genetic and epigenetic analysis of high-risk myeloma, and so we're looking for markers of high-risk disease and then trying to understand the biology associated with those markers to better target these patients that have really bad outcomes. So we analyzed the COMPASS data. One of the unique aspects of the COMPASS data set is this long insert sequencing, and it picks up structural variants and translocations. And so we looked at the translocations, and one, it provided a level of detail of all the translocations that really is unprecedented. One of the things that we found from this is that about 7% of newly diagnosed patients have translocations in the immunoglobulin lambda light chain. And these patients, they tend to do worse than other patients. And so there's something intrinsic to those patients with an IGL translocation that leads to bad outcomes. That's one part of the work that we're doing. The other aspect to it is we've graciously been granted specimens from the MMRF COMPASS study. And so we're now in the process of doing a different type of analysis where we look at epigenetic modifications. And so these are small chemical modifications to DNA. Specifically, in our case, we're looking at something called DNA methylation. And this is indicative of the chromatin state. And so this tells us a little bit about the regulatory potential of the cells. And so we're in the process of generating this data for hundreds of specimens, integrating it with the genetic and RNA analysis to understand a more complete and view of the molecular program of myeloma, and to understand if there are any epigenetic markers that may be informative in identifying high-risk disease.